Have you ever stopped to think about what's happening inside you right now? I mean, how your body just handles the food you eat,
Yeah.
turns it into energy or stores it away. It's pretty amazing, this invisible work.
It really is almost magical, isn't it?
Totally. And that's what we're diving into today, this incredible dance between carbohydrates and fats, how our body manages them together. We're using Chapter 34 of Marks Basic Medical Biochemistry, a clinical approach, as our guide.
Yeah, and our mission really is to shed light on that whole system, how the body switches, between storing fuel right after you eat and then tacking into those reserves when you're fasting.
Uh-huh.
And this isn't just some abstract idea, it's fundamental. It's how we survive, how we stay balanced and fueled.
Right. And pulling the strings behind the scenes, you've got these key players, hormones like insulin and glucagon, which are just crucial.
Absolutely critical.
And then there's the amount of fuel actually floating around in your system and, well, what your body needs right now. It's like a constant negotiation.
Exactly. A negotiation. That's a good way to put it.
Yeah.
So today, we'll explore how that all works. We'll look at how cells control their enzymes, the different pathways involved when you're fed versus fasting.
And the clinical side, too.
Definitely. We'll see what happens when this delicate balance gets disrupted. It really brings the biochemistry to life.
Okay, let's get into it. Before we talk about what happens to food, how does the body even know? When to store, when to release? What are the basic controls?
Well, it really boils down to controlling enzymes. They're the workers making everything happen. And our source material points to 4 main ways the body does this, kind of at different speeds.
Right.
So the fastest, like instant control, is allosteric regulation. Think of it like molecules binding to the enzyme, not at the active site, but somewhere else, and changing its shape. Click on, click up.
But dimmer switch, almost.
Sort of, yeah. Then, a little bit slower, but still fast, you have covalent modifications, mostly adding or removing phosphate groups, that's phosphorylation and dephosphorylation. That flicks enzymes on or off too.
Right, I remember that. Phosphorylation.
Then for changes that take hours, the body can actually change how much enzyme it makes. That's induction and repression of enzyme synthesis.
So making more or less of the worker enzyme itself.
Exactly. And finally, the longest term control is just degrading the enzyme, getting rid of it. But the really key thing is pathways aren't just one switch. They have multiple controls, multiple regulators. It allows for this fine-tuning, a graded response, So supply perfectly meets demand.
Makes sense, like having multiple checks and balances?
Precisely. Pyruvate dehydrogenase, PDH, is a great example. It's got several layers of control acting on it.
So with all these controls in place, what's the main conductor? What tells the body, okay, time to store or time to release?
Wow, that's where the hormones come in. The big two are insulin and glucagon. And the crucial thing is they often work in opposition, especially in the liver. Insulin says store, glucagon says release.
So they counteract each other.
They do. And it's really the ratio between them, the insulin to glucagon ratio, that dictates the overall metabolic direction. High insulin, low glucagon. Storage mode. Low insulin, high glucagon. Release mode. That ratio is paramount.
Okay, let's picture it. You've just had a meal, lots of glucose, maybe some fats coming in. Insulin is high. What happens? Where does all that energy go?
Right. So in this fed state, high insulin-glucagon ratio, storage is priority 1. Your liver gets busy making glycogen. It might have, say, 80 grams after an overnight fast, but after a meal, that can jump to 200, even 300 grams.
Wow, quite a bit.
Yeah. But once glycogen stores are topped up, the liver switches gears.
Yeah.
It takes that extra glucose and starts making fat triglycerol.
It converts sugar to fat.
It does. But it doesn't store it itself. It packages this fat into particles called VLDL, very low density lipoprotein, and sends them out into the blood.
Okay, so VLDL is like the liver's fat delivery truck.
Exactly. And where does it deliver? Mainly to your adipose tissue, your fat cells. And those cells, they have an almost unlimited capacity to store this fat. Abdomen, hips, thighs. That's where it primarily goes. It's this coordinated effort between liver and fat tissue to capture all that incoming energy.
So the liver is central. How does it manage that switch? Converting glucose first to glycogen, then to fat. What are the key steps?
Well, first, glucose enters the liver cell. An enzyme called glucokinase traps it by converting it to glucose 6-phosphate. Glucokinase is smart. It works best when glucose levels are high, like after a meal. Insulin also nudges it along by telling the cell to make more of it.
Okay, trapped glucose, then glycogen.
Right. The main enzyme for building glycogen is glycogen synthase. And guess what? High insulin activates it. Low glucagon activates it. It gets the signal. Time to build glycogen.
Makes sense. But what about turning glucose into fat? That sounds more complicated.
Wow.
It involves glycolysis, breaking down glucose. A key control point here is PFK1, phosphofructokinase 1. It gets a major boost from a molecule called fructose 2-6 bisphosphate. Think of F2006BP as a big green light saying, go, go, go with glycolysis. We've got plenty of glucose.
That name came up before, so it pushes glucose breakdown.
It really does. It ensures glucose flows down the pathway towards pyruvate, and then pyruvate needs to become fatty acids.
Right, and pyruvate's in the cytosol, but fat synthesis parts happen in the mitochondria. How does that work?
Good question. It's a bit of a journey. Pyruvate first goes into the mitochondria. There, pyruvate dehydrogenase, or PDH, converts it to acetyl-CoA. PDH is humming along when insulin is high.
Okay, acetyl-CoA inside the mitochondria, but you said fat synthesis is in the cytosol.
Exactly. Acetyl-CoA can't just walk out, so it does something clever. It combines with another molecule, oxaloacetate, OAA, to form citrate. And citrate can leave the mitochondria.
Yeah, the citrate shuttle.
That's the one. Citrate carries those acetyl-CoA carbons out into the cytosol. Once there, an enzyme called citrate lyase breaks it back down, releasing the acetyl-CoA. Now it's ready for fat synthesis.
Okay, clever trick. So now we have acetyl-CoA in the cytosol. What next?
The next big step is converting that acetyl-CoA into malonyl-CoA. The enzyme for this is acetyl-CoA carboxylase, or ACC. This is a major control point.
Why is it so important?
Because ACC is highly regulated, citrate turns it on a signal of abundant building blocks, insulin turns it on, but built-up fatty acids turn it off. And malonyl-CoA, the product, it has this critical dual role.
Dual role.
Yes. First, it provides the 2 carbon units needed to build fatty acids, specifically palmitate. But second, and this is crucial, malonyl-CoA acts like a stop sign for fat burning. It inhibits an enzyme called CPTI, carnitine palmitoyl transferase I.
CPTI, what does that do?
CPTI is the gatekeeper that lets fatty acids into the mitochondria to be burned for energy. So by inhibiting CPTI, malonyl-CoA ensures that the fatty acids just made are directed towards storage, not immediately burned. It's brilliant, really. Make fat and stop burning fat simultaneously.
Wow. Okay, so the body makes fat, packages it as VLDL, and stops itself from burning it right away. But how does that fat actually get into the storage cells from the blood? You mentioned VLDL and chylomicrons from food.
Right. Both those particles, chylomicrons carrying dietary fat and VLDL carrying liver-made fat, circulate. On the lining of capillaries, especially in muscle and fat tissue, there's an enzyme called lipoprotein lipase, LPL.
LPL, got it.
LPL basically chops up the triglycerides in those particles, releasing fatty acids and glycerol. Muscle LPL is always pretty active, grabbing fuel, but adipose LPL, it really ramps up after a meal and insulin is high. Insulin tells the fat cells to make and secrete more LPL.
So insulin helps fat get stored.
Directly. The released fatty acids enter the adipose cells. Now, to store them as triglycerides again, they need a glycerol backbone. Adipose cells can't really use the glycerol released by LPL. Instead, they rely on glucose coming in again, thanks to insulin-boosting GLUT4 transporters to make that glycerol backbone.
It's also connected. Insulin seems to be everywhere in this fed state.
Absolutely. It's the master hormone of storage.
So connecting this back, you mentioned those clinical examples from Marks. How does this explain their situations? Like Connie C.
Right, Connie C, with her insulin-secreting tumor, her body was basically stuck in fed mode all the time. Constant insulin signal meant constant fuel storage, glycogen, fat, which explains her weight gain. Her system couldn't easily switch to release mode.
And Dianne A and Deborah S, the ones with high triglycerides.
Yeah, hypertriglyceridemia. Dianne, type 1 diabetes, virtually no insulin. Without insulin, her LPL activity was way down. She couldn't efficiently clear fats, chylomicrons, and VLDL from her blood.
So the fat just built up?
Exactly. And Deborah, type 2 diabetes, had insulin resistance, particularly in her fat tissue. So even if she made insulin, her fat cells weren't responding properly to make enough LPL. Same result. High triglycerides, that opalescent look to her blood serum they described. It really shows how vital proper LPL function, driven by insulin action, is for clearing fats.
Okay, that paints a clear picture of the fed state. Now let's flip the coin. Hours have passed. The meal's energy is gone or stored. You're fasting. How does the body keep everything running, especially the brain?
Good question. Now the insulin-glucagon ratio flips. Insulin drops, glucagon rises. The liver becomes the hero again, but this time for producing glucose.
How does it do that?
2 main ways. First, glycogenolysis. Breaking down that stored glycogen. The high glucagon signal activates enzymes, particularly glycogen phosphorylase, that start chopping up glycogen, releasing glucose into the blood, and it simultaneously turns off glycogen synthase, the building enzyme.
Okay, using of the stores first.
Right, but those stores only last so long. So the liver also ramps up gluconeogenesis, making glucose from scratch, basically, using things like amino acids from muscle protein breakdown or lactate.
Making new glucose. How is that controlled?
Key enzymes for gluconeogenesis get synthesized more. And remember, fructose 2-compyl-6-bisphosphate, the green light for glycolysis? In the fasting state, its levels plummet. That acts like a red light for glycolysis and simultaneously removes the inhibition on gluconeogenesis. The liver switches from using glucose to making it.
A very deliberate switch.
Very. Driven by that falling insulin-glucagon ratio and signals like CCAMP activated by glucagon.
So thinking about Dianne and Deborah again,
Yeah.
how does this fasting state look different for them?
Well, it's kind of like their bodies are stuck in an exaggerated fasting state, metabolically speaking. Dianne, with no insulin, can't get glucose into her muscle and fat cells properly. Plus, her high glucagon drives her liver to pump out tons of glucose via glycogenolysis and gluconeogenesis. Result, severe hyperglycemia.
Dangerously high blood sugar.
Exactly. Deborah, with insulin resistance, has a similar problem. Her tissues don't respond well to insulin, so glucose uptake is poor, and her liver might still be overproducing glucose because it's not getting the stop signal from insulin effectively. Also leads to hyperglycemia.
Okay, so the liver's making glucose. What about fat tissue during fasting?
Fat tissue switches from storage to release. The low insulin and high glucagon and other hormones like epinephrine trigger lipolysis fat breakdown. Enzymes like adipocyte triglyceride lipase and hormone-sensitive lipase, HSL, get activated.
The HSL, I've heard of that one.
Yep. They start snipping fatty acids off the stored triglycerides. These fatty acids are released into the blood, bound to albumin, and travel to other tissues like muscle and the liver to be used as fuel.
So fat becomes the main fuel source for many tissues.
Increasingly, yes, especially during prolonged fasting. Those fatty acids go to the liver. Now, because malonyl-CoA levels are low, remember ACC is off, CPTI is active, letting the fatty acids into the mitochondria.
The burning.
For beta oxidation, yes, burning fat. This generates a lot of acetyl-CoA inside the liver mitochondria. But the liver often produces so much acetyl-CoA that it can't all go into the TCA cycle immediately.
So what happens to the excess acetyl-CoA?
The liver converts it into ketone bodies, acetoacetate and beta-hydroxybutyrate mainly. These ketone bodies are then released into the blood.
And other tissues use these ketone bodies.
Exactly. Muscle uses them readily. And importantly, after a few days of fasting, the brain adapts to use ketone bodies as a major fuel source. This is crucial because it spares glucose. It reduces the need to break down muscle protein just to get precursors for gluconeogenesis.
So ketones are actually protective in a way, sparing glucose in muscle.
In normal fasting, yes, they're a vital adaptation. The high acetyl-CoA also has feedback effects. It inhibits PDH, stopping glucose from being turned into acetyl-CoA, and activates pyruvate carboxylase, pushing pyruvate towards gluconeogenesis. It all fits together to prioritize glucose production and fat ketone use.
But you mentioned they can be dangerous, ketoacidosis.
Right, that's where the balance breaks. Dianne A, with severe type 1 diabetes, is the classic example. Her lack of insulin means lipolysis runs rampant. Massive amounts of fatty acids flood the liver, leading to runaway ketone body production. These ketone bodies are acidic, and their massive accumulation overwhelms the body's buffering systems, leading to diabetic ketoacidosis, DKA. It's a medical emergency.
Why isn't it as common in type 2 diabetes, like with Deborah?
It's interesting. The thinking is that even with insulin resistance, people with type 2 diabetes often retain some insulin production or sensitivity, especially in fat cells. Just enough, perhaps, to put a partial brake on HSL and prevent that completely uncontrolled massive release of fatty acids. So they get hyperglycemia, but usually not full-blown DKA. It's a key difference.
Fascinating. And what about muscles during all this, fasting or exercise?
Muscles adapt too. During exercise, they first burn their own stored glycogen. Then they take up blood glucose. The liver keeps supplying that glucose. As insulin drops, you'd think glucose uptake would stop, but high levels of AMP inside the muscle cell, a sign of low energy, activate a key enzyme, AMP-activated protein kinase, or AMPK. And AMPK can actually stimulate glucose uptake independently of insulin, especially during exercise.
So AMPK provides a backup way to get glucose into muscle.
Exactly. But as exercise continues and those fatty acids are released from adipose tissue, muscle starts burning them too. The products of fat burning then actually slow down glucose use in the muscle, helping to spare blood glucose for the brain. It's another layer of fuel management.
It really is an intricate dance. You mentioned AMP and fructose 2.6-bisphosphate acting like master switches. Can we zoom in on those?
Absolutely. They are incredibly important signals. Let's start with AMP. It's basically the cell's low fuel warning light. When ATP is used, you get ADP. But an enzyme called adenylate kinase converts two ADPs into one ATP and one AMP.
Okay.
What this means is that even a small drop in ATP can cause a much larger percentage increase in AMP. So AMP levels shoot up dramatically when energy is low.
A very sensitive gauge.
Extremely sensitive. And high AMP is a powerful signal. It activates pathways that generate energy catabolic pathways, often by activating that AMPK enzyme we just mentioned. Conversely, when energy is high, AMP drops, ATP rises, and that favors energy storage anabolic pathways.
So AMP levels directly tell the cell whether to burn or build.
In a nutshell, yes, it's a fundamental energy sensor. And then there's fructose 2,5,6-bisphosphate. We talked about it being the green light for glycolysis in the liver.
Right, turning on PFK1.
Exactly. When glucose and insulin are high, its levels are high. This pushes glucose breakdown, glycolysis, and simultaneously inhibits glucose synthesis, gluconeogenesis. But when glucose and insulin are low and glucagon is high, its levels drop.
And that flips the switch.
Completely flips it. Low F2006BP inhibits glycolysis and allows gluconeogenesis to proceed. So it acts as this crucial decider in the liver. Are we breaking glucose down or are we making it? It's controlled by the very hormones signaling the fed or fasting state.
These small molecules have such huge impacts. Okay, let's dive a bit deeper into AMPK. You said it's activated by AMP, low energy. What exactly does it do once it's active?
Right, so AMPK acts like the cell's financial manager during hard times. When energy is low, high AMPK gets activated, and its main job is to restore energy balance. It does this by shutting down non-essential energy spending.
Like what?
Things like making fatty acids, it inhibits ACC, making cholesterol, inhibits HMG-CoA reductase, making triglycerides. Even protein synthesis gets turned down, partly through its interaction with another pathway called mTOR. Basically, any process that consumes a lot of ATP gets put on hold.
Okay, so it cuts spending. Does it boost income?
Yes. At the same time, it ramps up energy-generating processes. It promotes fatty acid oxidation, burning fat. It helps with glucose uptake in muscle, as we mentioned. It basically coordinates this whole shift towards generating ATP and conserving energy.
It sounds incredibly central. How is it activated so precisely? Just by AMP?
Well, AMP binding is key. It makes AMPK more susceptible to activation. But it also needs to be phosphorylated by upstream kinases, like LKB1. These kinases themselves often sense energy stress. So there are multiple inputs ensuring AMPK responds appropriately to the cell's energy status. It gets turned off when energy levels recover and AMP drops.
So it's not just on-off, but finely tuned by these multiple signals.
Exactly. It's a highly sensitive system designed to react quickly and appropriately to changes in cellular energy. It really highlights how integrated everything is, energy levels directly controlling the machinery for both fuel use and storage.
What an incredible journey. We've really traced how the body manages carbs and fats, switching between storing after a meal and releasing during fasting. It's this amazing, complex dance.
It really is. We've seen the immediate enzyme controls, the slower hormonal shifts with insulin and glucagon, and these key intracellular signals like AMP and fructose 2,6-bisphosphate acting as crucial switches. And understanding this integration is key to seeing why problems arise in conditions like diabetes or hypertriglyceridemia, like in those clinical cases.
Yeah, from packing away glucose as glycogen and fat as VLDL when you're fed, to releasing glucose from the liver and making ketone bodies when you're fasting. It's all connected to keep your cells fueled.
And learning about these pathways isn't just memorizing names, it's about appreciating the sheer elegance of the biochemistry inside us. It gives you that foundation to really understand metabolic health.
Definitely. So maybe next time you finish a meal or feel hungry later, think about this. How is your body adjusting? How might subtle changes like exercise or diet be tweaking those master regulators like AMPK? It really makes you appreciate the constant invisible work going on inside.
It certainly does.
Thank you for joining us on this deep dive into carbohydrate and lipid metabolism. We hope you feel a bit more informed and maybe, like us, a little more amazed by it all.